Sounds like it. All right, for anybody who might not know me, I'm Paul Orchard. I'm in the Department of Peds, but in the Division of Bone Marrow Transplantation. And so every year we have the Crivet Lectureship. So for those of you that don't know Bill, and Bill's been dead for almost 15 years now, I'd just like to mention a little bit about why we honor Bill and what his contributions were. So Bill also was in hematology, oncology, and bone marrow transplant. He was the department chair for about 10 years or so, but was a very innovative thinker and a great scientist, and published important works in coagulation and hematology and a number of fields. But his primary interest was in transplantation and transplantation for rare metabolic disorders. So, Bill published a number of very important papers in terms of making a big splash in the New England Journal and Lancet, and I've got a couple of these papers here. So he really helped establish the University of Minnesota as one of the go-to places for transplantation for these rare disorders, and what seemed to work and what didn't seem to work and why it didn't seem to work. So we're still, of course, interested and spend a lot of time thinking about these disorders and seeing a lot of these patients and trying to figure out how to move the field forward, and that's the impetus behind the Crivet Lectureship. So today I have the opportunity to introduce Keith Van Haren, who's here from Stanford. So Keith was originally in the Grand Rapids area, and then trained in Rochester, so where'd you do your, Mass General, right, is where you did your residency? And then went to Stanford and has been at Stanford since that time. So he's an expert in adrenoleukodystrophy, which is one of the diseases that we're very interested in. And without any further ado, Keith.
Thank you. So, yeah, thanks for inviting. It's a pleasure to be here. I never met Bill, but everyone in the field of transplant and also pieces of the field of neurology know Bill's impact and the influence he's had really globally in treating these rare disorders. It's a remarkable legacy. I don't have many disclosures that are really relevant to this talk. I'm not talking about any specific therapies in much detail, but I do some consulting. And some of my research, which is alluded to here, is funded by the NIH and some foundation work. So here's a quick overview of what we'll be talking about. What I'm going to do is introduce the idea that some of these heterogeneous complex disorders can shed light in maybe a bidirectional fashion with our rare monogenic disorders. So in the case of adrenoleukodystrophy here, particularly this is kind of a new conceptual map for cerebral demyelinating adrenoleukodystrophy. The idea here is that multiple sclerosis and cerebrovascular stroke may have some unexpected insights for cerebral Ald and probably vice versa. Okay, so here is the first histologic description of adrenoleukodystrophy. You'll notice the term they applied was acute multiple sclerosis. We're describing kind of how we don't know what causes multiple sclerosis, and that is still true today. But this is the reality for many of the clinical syndromes we see, right? As they begin as a kind of a collection of similarities, and we gradually separate them out into distinct entities over time. And this is what's happened to adrenoleukodystrophy. It was considered for a very brief period of time part of multiple sclerosis, and now, as we know, is something completely distinct, or so we think. But what do we know about ALD? So we know it begins with a mutation in the ABCD1 gene. This provides a function of transport of these saturated fatty acids into the peroxisome where they're normally broken down. When this gene doesn't work, these fatty acids build up and well, then we're not exactly sure what happens, but at least on a molecular level, believe it or not, but we understand what happens on a clinical level, an increasing level of detail. So we know that ALD varies by gender. This is just classical X-linked physiology at work. So males have the phenotype that we know predominantly, but of course women do also have a phenotype. It's just a milder, later onset form of the disease with a narrower set of symptoms. And the challenging and poignant part of this disease is that every boy begins essentially looking perfectly healthy for at least the first few years of life. And it is only later in life that we often discover, in the absence of newborn screening, that a child is affected. And we can discover this through a variety of ways, sometimes through adrenal insufficiency, sometimes through cerebral disease, and in some cases, men make it all the way into adulthood, and women too, and then present with some symptoms of walking difficulties and so on that we then discover are related to the disease. So adrenal insufficiency increases over the course of the lifespan. This thing that we call adrenal myeloneuropathy, which is a long name for really what is probably best conceived as an axonal disorder of central axons and peripheral axons. So it's basically a spastic paraplegia syndrome and a neuropathy syndrome combined. And I think you can think of this as the default form of ALD. Everybody gets this. If you live long enough, you'll get this. And I think of this as like an accelerated form of aging of the spinal cord and the nerves. So it basically accelerates. So if you take the stereotype of the aged man or woman who walks with an increasing slouch and eventually is using a cane, same idea here. It just happens in your 30s or 40s or 50s. So it happens much earlier. And then there's the form we'll be spending the rest of the talk discussing today, and this is the cerebral form of ALD. So this is cerebral demyelinating ALD. This is the thing that looked like multiple sclerosis in that first slide I showed you. And this is the kind of most poignant and unique and potentially challenging aspect of the disease, certainly in childhood. There's one piece I want to make clear at the outset here, and that not everybody gets this form of the disease. Everyone gets the spinal cord form of the disease, and most get the adrenal form of the disease. But not everyone gets this form of the disease, and we don't know why. We also know that there is your biggest risk factor for developing cerebral demyelination is your age. And your peak risk is in the first decade of life, specifically between the ages of 3 and 8. We'll come back to this point, but I want you to remember this. And so there are kind of a bimodal risk here. So the highest risk is in childhood. There's a later onset risk in adulthood. But about a fifth to a third of patients never get this. And this is an important point we'll come back to. So let's focus on this a bit more. So what more do we know about cerebral ALD? Well, we know it typically begins in one of a couple of areas. It begins typically in the densest part of the fatty portion of the brain, the myelin. So in that the densest area is actually right in the corpus callosum here, at the back of the corpus callosum, the front of the corpus callosum. This is where the disease begins in the vast majority of boys. And it begins as a small lesion, and right in the middle of the brain, and that lesion gradually grows outward. It's a pattern that's somewhat reminiscent of a glioblastoma multiforme, the GBM tumor. Begins in a similar place, a similar pattern, spreads outward. Here's this annualized risk curve. Just to highlight this again, we know that boys around the age of 3 or 4, and between 3 and 8, this is their highest risk of disease. So annualized incidence is reasonably known. And these lesions grow outward. They become, these boys go from healthy, no lesion here on the left, to when these lesions develop, they still have no symptoms, and they actually have no symptoms until lesions are reasonably large. So this is an MRI from a boy who had recently become symptomatic and had gotten an MRI that diagnosed him, and he had really advanced disease and was no longer qualifying for transplant. So he presented in an advanced state. We know that these lesions carry a specific histological profile, and in one sense, these lesions are time capsules for a disease process at different stages. And so depending on where you look at the lesion, we can, if we do it properly, understand what's happening in different states and points in time for the disease. So let's say if we take this MRI here, extract out here, and we say this is kind of the normal appearing part of the myelin and gray matter, this is the edge of the lesion, and then this is the deeply affected part of the lesion where everything has already come and gone. We see some scattered activated microglia. These are the brain's immune cells. We see lipid-laden macrophages, so basically immune cells that are full of fat trying to digest it. We see then scattered T cells, B cells, and monocytes, and then we see complete loss of myelin and axons. Devastated brain tissue that never recovers. We know what happens if these lesions are left to their own devices. The lesions will continue to grow almost invariably. They grow steadily. The boys eventually will succumb to a vegetative state and will pass away from secondary causes, pneumonia, other complications. So it is not a happy picture. But something remarkable happened here at the University of Minnesota, really first, and that was that bone marrow transplant was piloted by your stem cell transplant group. And this is one of the first reports of a successful outcome. There had been tried unsuccessfully in advanced disease, but this was Bill Krivet's paper describing the first kind of early case series. And this is not from the same paper, but same data set showing a remarkable effect. This is the kind of treatment effect we all hope for in a clinical trial, right? We go from, something that is clearly devastating to a really pretty clear treatment effect within a few years of initiating the therapy. But there's a caveat here, and that's that you need to begin treatment when the lesion is still small. If you begin treatment when the lesion is large, you're lost. And here's the other twist. We don't really know why this works. This was one of the Hail Mary passes was thrown because we knew there was inflammation, and well, if it's inflamed, why don't we try transplanting it? This was after they'd tried a whole bunch of other anti-inflammatory strategies, which all failed. Bone marrow transplant worked, but we really don't understand why, and part of the reason we don't understand why is we don't really understand what initiates these lesions. In other words, why does a lesion form in some boys and not others? And then why do these lesions persist? Why does a metabolic disease like ALD lead to such profound inflammation? 40 years later here, we're still left without good answers. Well, meanwhile, because we have a definitive early stage treatment that requires early diagnosis, and we have diagnostic paradigms that can detect children at birth, states have begun newborn screening for this disorder. So New York started in 2013, and California and Minnesota and other states have since picked that up. We're now screening in quite a few states across the country. And this has had an interesting kind of off-target effect in that we now have families in our ALD communities who are in the haves and the have-nots. And the haves are, of course, depending on your vantage point, you are lucky if your child is detected through newborn screening. It doesn't feel that way on the day you get that news, but that is in a sense what is happening is you're moving into a fortunate paradigm. The have-nots are those boys like the one I showed you earlier who was presented at an advanced stage and was not treatable. So this splits up a little bit. We know that there are, as I mentioned, some boys that never develop lesions. and other boys that do. So the have-a-lots are those who never do, they just skip that part of the whole disease. They never get brain inflammation. And there are others we detect early. They have to go through a bone marrow transplant, which as we know, is not a gentle process, and it has some long-term sequelae, even despite the neurologic effects. And some of the have-nots wind up here too. We sometimes catch these boys early enough. But then there are these boys that are too late for transplant, and these are boys who've just missed the boat. And there's a particular tragedy in this paradigm where you've just missed something. There's a special poignancy of loss and a sense of deep regret and guilt and anger that exists here. When you know you could have something, you could have had something if only this, that, or the other. We can change this if we understand a bit more. If we understand the mechanisms of disease perpetuation better, this gives us the opportunity to help stop and arrest this disease. We think that this disease is arrestable. In a small number of cases, it stops on its own. It suggests there's some kind of homeostatic mechanisms at work. And to take it back even further in a more hopeful pattern, all these boys who are now detected on newborn screening, we've got time and we've got resources. We can really begin to think about this from a preventive standpoint. This is now potentially a disease with a preventive angle to it, much like any of our other, heterogeneous disorders of like, well, we know we need to find some risk factors. We need to think about things rationally. Can we implement safe and reasonable low-risk treatments that can move high-risk boys into low-risk categories? So here's how our lab is approaching this, and part of my role here is trying to encourage other people to think this way too, is to use our monogenic disorders, insight from these disorders, to try and match them with other disorders that resemble them. And we're starting here, I think, with the most obvious one for cerebral, which is comparing cerebral ALD to MS. So I think here's the tree I put together of myelin disorders. But you get the idea here. It's obviously not complete, but we have some genetic disorders in this group, and we have some acquired disorders. And ALD sort of crosses this bridge, and it bears a meaningful number of resemblances to MS. And so this is how our lab is conceiving the landscape. Is we can think about risk factors and then mechanisms. Maybe we can think about common therapies. These disorders, as I'll show you, bear so many similarities, there probably is common ground here. And so there are radiologic similarities to these lesions. This is not very meaningful, but it's a start. There are actually a lot of histologic similarities. So this is from a review that, sorry, it's not cited here, but it's a nice review describing the histologic features and differences of a few different demyelinating disorders. And believe it or not, ALD is really most similar to MS, more so than some of the other more classical childhood demyelinating disorders. But there are only a couple features that distinguish it, and one of this is this little thing here that I'll come back to, these needle-like inclusions in the macrophages that make ALD macrophages kind of distinct. So here's a cytokine profile. This is from data from your hospital here, collected by Dr. Lund and Dr. Orchard from a cytokine profile in the CSF compared to MS and control patients. And you don't have to squint too much to see some meaningful similarities here. You can see kind of a similar area of downregulation of cytokines in this realm, and then an upregulation of some of these cytokines here, which I'll come back to briefly. Particularly IL-1 beta, IL-18 is upregulated, and a few other specific antibodies are upregulated. So we, in a nutshell, jumped in and said, well, okay, if ALD looks enough like MS, multiple sclerosis in other respects, let's think about what things we know about MS. We know that, among other things, vitamin D is a risk factor. Vitamin D status is a risk factor for MS. The latitude you're born at, the month you're born, your vitamin D status late in life, these all predict your risk of MS. And so let's look at this on ALD. And we did in a retrospective cohort from Kennedy Krieger. And early life vitamin D status in these boys seemed to predict later life cerebral outcomes to a much larger effect than we expected. But I didn't understand why. You know, this is In MS, vitamin D is conceived as an immune attenuating, or vitamin D is a kind of immune modulating, immune tolerizing. And that could be true in ALD, but we're really talking about a primary metabolic disorder here. So what we thought is that there are some metabolic features to vitamin D's mechanisms that are probably underappreciated. And so this has been a big focus of our lab for the past couple of years, is trying to unravel this. And so we asked the question, well, really, if we ask agnostically, what does vitamin D do? What would the answers be? And so what we conceived of was just try and find human clinical trial samples, so blood samples from vitamin D trials. And we started with a clinical trial in MS and generated our own transcriptomes from these trials. We sorted into monocytes, B cells, and T cells, thinking that we might see a stronger signal in monocytes for various reasons. And we found something that we thought was very interesting. So we validated this same signal in a second cohort of non-MS patients. So there were patients with prediabetes who were otherwise pretty healthy. And we found something very interesting. We found that vitamin D exposure was flipping the immune cell profile from fat to glucose. We thought we might see a little hint of this, but we saw a lot of this, and this was pretty unexpected for us. It turns out that there's a growing literature around this. A small but growing literature. But we're specifically seeing kind of a downregulation of actually peroxisomal pathways and fatty acid elongase pathways associated with vitamin D exposure, at least on the transcriptional level. And we're looking at this in some functional levels now. And we're seeing an increase in glucose metabolism. And this is all immune cell derived. So we can only speculate as to how much this is relevant to brain and other tissues. But this is how the immune system is seeing this. And then we saw something else was quite interesting. We saw actually some signs that there was an energy efficiency gain. So mitochondrial oxidative phosphorylation transcripts were way down. Apoptosis signaling was way down. And we can see this functionally. This is pretty clear when we do functional assays just in vitro. This is one of the clear effects of vitamin D exposure just in a test tube with immune cells. And so vitamin D is keeping the immune cells alive and presumably happy. And they seem to have some energy-saving features at work here. And then, no surprise, we found a downregulation of immune transcripts. And so this is, I think, pretty well established at this point. We moved then into a kind of a causal. We generated the first Bayesian causal network for vitamin D and human immune cells for vitamin D at all. And we looked at this from a few angles. This is the most resolved angle we have, where we can see all the different gene set modules and where they are and what's moving. We see that vitamin D its first order effects appear to be metabolic. In other words, these are the first things that vitamin D is moving. It's moving fat metabolism down, including via cholesterol homeostasis, and it's moving glucose metabolism up, at least on a transcriptional level. And then the downstream effects are, you can see in, so decreased peroxisomal biology, decreased adipogenesis, and then a strong decrease in oxidative phosphorylation transcripts, suggesting potentially an energy savings, decreased free radical reactive oxygen species, and then the expected changes in immune profile, so a significant downregulation of immune signaling. So this is pretty interesting. This is now a mechanism that we did not have, or plausible mechanism we did not have before, where we're moving pieces of biology that appear to be specifically relevant to ALD with vitamin D exposure. Is this potentially how vitamin D exposure might be de-risking the risk of cerebral ALD? I don't know that there's one single thing I can point to in here that says yes, but there are several pieces that are pretty suggestive, including the fact that we're hitting some peroxisomal targets potentially favorably. So let me switch gears now to another disease and see where we can go with this. So this is a little less intuitive. What if we compared cerebral ALD to cerebral vascular stroke? Now, let me set some background here. This is actually one of the most important slides I want to show you, and this is what I call the neurometabolic hurdle of childhood. I think many people are unaware of this really fascinating phenomena, but most vertebrates spend about 3 to 5% of their energy on the brain. There's a term for this. It's called encephalization, how much of the body's energy gets expended on the brain. The reason there's a term for it is that humans do it very uniquely. And so mice do about 4% of energy expended on the brain, chimps about 13%, adult humans are about 20%. Toddlers spend about 2/3 of their basal energy on brain development. There's no other comparison in the natural world that does this. So one way to look at childhood is the first decade of childhood is that it is really dedicated to fueling the brain. And one way to look at human evolution, our evolution from our non-human primate and non-primate relatives, is that human evolution has really been focused on engineering the system to basically grow the brain at great metabolic expense. And so this has put us in a whole bunch of unique predicaments as a species. We're out kind of on a very distant evolutionary limb. We're doing something that no other species is really modeling. And this is what makes it hard to study in the lab. And it's a good reason to think about single gene disorders as one, maybe perhaps one of the only ways to really understand these things. Let's take then get another look at our cerebral ALD risk curve. Remember I told you that between the ages of about 3 and 8, we have a peak incidence of cerebral ALD. And look what happens when we lay these two over the top of each other. They really resemble each other, remarkably. And in the world of metabolic diseases, especially neurometabolic diseases, this is what we typically say. We say, well, we see these disorders present during periods of peak metabolic demand. We just, for whatever reason, we haven't really thought of ALD in quite the same way, but we can now. I mean, I think this is, it looks like kind of a classic metabolic failure. We have a potential, what this looks like is a supply-demand mismatch. That may be part of what is responsible for this spike in incidence in childhood. Is we've got a huge demand, and some boys are not able to generate the supply, whatever that might be, whatever supply of energy the brain needs at any one point, whether it's some period of infection that's pulling energy away from the brain, et cetera. But this is probably true of lots and lots of childhood disorders. This is absolutely not unique to ALD. This is true of lots of childhood disorders. The brain asks for a lot, and we have to. The body is in triage during childhood, put in a difficult situation much of the time, particularly during infection, when many of the things that in human evolution have been triaged to later in life have been triaged: growth, sexual development, strength, advanced cognition. But you can't triage immune defense. You have to maintain that through childhood for the survival of the organism. For the survival of the child. The immune system must be fed during periods of time. It's one of the many things that must be fed. But this is sometimes puts it in competition with the brain. So perhaps we're onto something here. So this is in keeping with some work from Patricia Mussolino at Mass General Hospital, who is a critical care neurologist who took an interest in ALD. And, of course, viewing it from a different lens, she was looking at the perfusion and vascular markers in ALD brains, and she said, this is not normal. These brains look like they're hypoperfusing, like an early stroke. And so she's pursued this angle to great effect, has found some really fascinating insights here. But and I think this matches up very nicely with what we're seeing here. Basically, stroke is a supply-demand mismatch. We just do it as in adults by a kind of a very abrupt clotting fashion. But there's something more subtle happening here. Oops, right here. So what if we, instead of thinking that this is MS, we looked at stroke and compared these two disorders? So I've started thinking about this from actually a slightly different tangent that won't make perfect sense here. So let me explain what another investigator, basically a stroke biologist, was investigating, and he and I met on a common note. But He did not have any interest in ALD until a few months ago, when I reached out to him and started telling him what we were saying. But he had been looking at the chronic effects, basically the post-stroke inflammatory signal, which, surprising to me, really is totally not on the radar of neurologists or stroke biologists. He was really pioneering this, a few years ago, looking at, well, what happens if you induce a stroke and you just watch for months at a time? What happens? So this is what he did. It turns out that strokes get a huge amount of immune inflammation. They liquefy. They get huge lipid-laden macrophages, enormous immune deposition, a lot of activity, leaky blood-brain barrier. It looks not unlike, I won't draw too many analogies here, but it looks not unlike what we're seeing in cerebral ALD to an extent. We haven't examined this in granular fashion yet, but this is very analogous. And so he had set up some really interesting electron microscopy here and is taken a specific interest in foamy macrophages, which we also see in cerebral ALD and MS. And he's finding these cholesterol crystals. Remember, this is the thing that I said distinguished, that this was highly prevalent in ALD lesions, less prevalent in MS lesions. But this has been one of his key interests. And in fact, this is actually what got me interested before I even began thinking about it in stroke. One of the principal problems we have in our in vitro models of ALD is that we can't get these very long fatty acids into solution. They're stuck in solid form. And so we're fretting with this from just like a practical end of, I can't get this into solution. I want to get this into my cells so I can test it. But it occurred to me that and it actually kind of winds up in a crystal form. And it occurred to me one day that the cells probably have this very same problem, is they want to keep these fats in solution state too, because there is a paradigm here where cells are not just threatened by infectious agents, right? They also have to contend with physical threats, just like we do. And among those physical threats are things like crystals that actually have real sharp edges that can poke holes in their membranes and pose a physical threat to the cell and to the tissue. And so it is one of the challenges of a cell and of the organism to keep these things in solution, or if they're not in solution, quarantined in some vacuole until it can be properly disposed of. And this is part of kind of what they call innate immune kind of motifs. So there are a few varieties here, but this is kind of what we call a DAMP pathway, a danger-associated molecular pattern. In other words, our cells, all immune cells, and kind of throughout biology are designed to recognize specific motifs, among them crystals. And the reason is that these crystals, in part, pose a physical threat to the cells. And so we have entire molecular mechanisms dedicated to this task. Among them, and probably the most famous, is this thing called the Nlrp3 inflammasome. There are different portions of this thing called the inflammasome, but this one in particular is of interest because it is the thing that has been studied the most in beginning with gout more than a decade ago, and more recently with cholesterol accumulation and atherosclerosis. This is what drives inflammation and atherosclerosis, these cholesterol crystals. Really fascinating stuff. And it turns out that one of the primers for this system is our free fatty acids, saturated fatty acids, and combined with cholesterol crystals. And we know that this is one of the setups as part of ALD biology, and Christian Doyle, the stroke scientist at the University of Arizona, is studying this in a classical stroke model, and this is what's catching his attention, is this mix of cholesterol crystals in these macrophages that appear to be driving this Nlrp3 inflammasome. So in his model, he sees a little bit of basically cholesterol crystal deposition in the first month, more at 8 weeks, and here's basically he sees basically no cholesterol crystals in the stroke lesion at 24 hours, none at 1 week, but at 4 weeks and then at 8 weeks, it's increasing. So more cholesterol crystals are appearing, and in that same time frame is this particular cytokine, IL-18, interleukin 18, which is kind of reasonably specifically associated with this that appears at the same time. And so here's his model. I won't delve into it too much, but it's basically that these crystals are activating the inflammasome in the stroke lesions. And so we had begun looking at this before we met Christian, but and this is not a totally new idea that cholesterol esters are part of the lesions in ALD. It's been actually described by, many years ago, but we were looking at it from a new light. And so this is a really interesting view. I thought, well, okay, we'll look at these lesions again and see what kind of burden we see for these cholesterol crystals. So this is basically under a light microscope an ALD lesion, and this is when you twist a polarizing lens beneath it, birefringent crystals light up. And this is, I'll tell you, an extraordinary burden of cholesterol crystal deposition in this tissue. So again, using some of the spinal fluid from that was collected here at the University of Minnesota, we looked at specifically IL-18 cytokines in a large number of boys. We're clearly seeing an elevation in the spinal fluid of these boys during active cerebral disease. And so if we look at a larger profile here, and let's see how far does this comparison run with stroke. Again, not a lot of squinting required to see that these motifs here, TNF and some of these other IL cytokines are also downregulated here, and we see an increase in IL-18, IL-8, IL-7, and so on in our ALD patient. So there are some meaningful similarities here, and this is I didn't include a lot of data here. This is Christian's unpublished data, but he's building this really fascinating story around this molecule here, cyclodextrin. Many of you may know this molecule as it's basically a drug packaging agent. It's on the GRAS list for pharmacologic meds, so generally regarded as safe for use in just about anything. It's basically a chaperone that's used to solubilize lipids. So if you've got a lipophilic molecule that you want to deliver in a drug, you basically put it in your packaging material and it helps get it to target. And it's shown up in a couple of disorders because it was in the placebo arm, and the placebo was as effective as the treatment, but more effective than natural history in some of the models. And this is true of I think Gaucher and perhaps Niemann-Pick is maybe how they became interested in it. But it actually has been explored in atherosclerosis as a cholesterol mobilizer, and in Christian's stroke model, it's looking fascinating. It really is having a meaningful impact. He's using very high doses of it, but it's having a really meaningful impact on lipid and cholesterol clearance. So for him, and he's thinking of it as a post-stroke treatment. We in the ALD community are wondering if something like this, what's considered, within quotes, reasonably safe, at least at the doses it's used for drug packaging, as a possible therapy for advanced stage disease, specifically targeting something like cholesterol crystal formation, fatty acid crystal formation, and inflammasome biology. So where will this go? I don't know yet, but we can be hopeful. And I think there are some other obvious tangents here. We have no mouse model for cerebral ALD. And I think Troy and Paul and I were just talking last night about the possibility of thinking about basically using a classical stroke mouse model and applying, just inserting an ALD mouse and waiting. Perhaps it won't do exactly what we expect. It may not produce a permanent evolving lesion, but we may have some markers here that we can use, cholesterol deposition, fatty acid, associated inflammation, et cetera, to use and see if it works perhaps a little bit more specifically for our particular disease. And I should add that there are other companies that are interested in advanced cerebral disease, not exactly from this angle yet, but there are a few reasons to be hopeful that we're moving towards some meaningful insights to fill in that black box. So in summary, I think this is kind of the key thought here, that kind of the larger picture thought is that rare monogenic disorders may model focus pieces of common heterogeneous disorders. And I think this is very much the same as we try to listen to our patients and our families. I think that really, without projecting too much of our preconceptions on exactly what's happening to them, and really good clinicians know how to do this intuitively. They listen to their families and they say, well, this isn't exactly the way I would characterize this disease, but maybe there's something in here that this parent is telling me about what's happening to the child that I need to understand in a different light. And I think that as scientists, we need to be doing this in the same way. And as physician scientists, same way with the biology. If we have too many preconceptions about the state of the natural order of things, we will miss all these important clues. And if we alternatively allow to make some of these tangents to disorders that don't seem necessarily intuitively related at the outset, stroke and cerebral Ald, we may find some really important things, not just for the rare disease we're interested in, but for basic human biology and common heterogeneous disorders. My MS colleagues who study vitamin D on one side and my stroke colleagues who study stroke inflammation on the other are both very interested in what we're looking at in ALD. I'm very interested in their insights from their disorders. They're very interested in ours. And I think this is really fruitful ground where these disorders are going to inform each other. So if we start with the principle of like ALD is its own thing, let's just focus on understanding this alone. If we actually broaden our view a little bit and we think, we've got this climbing route here. We're going to try and climb it, but maybe there's if we think and look a bit to one side, to our left, and say, well, okay, actually, I see some similarities here. Maybe we can cross over to this adjacent disease, multiple sclerosis on our left, and try and glean some insights. And maybe there's some similarities here in stroke. We can cross over here and see if we can look backward onto our disease, and vice versa. They gain some insights here. And we can then begin to think about some specific avenues of shared biological space that may lead us to some really interesting places that we didn't expect to be. And I think this is particularly true of neurologic disorders because neurobiology in humans is so distinct from the neurobiology of mice and other model animals. There are pieces of this that we will never get at. We will never get definitive knowledge of human neurobiology through a mouse. And I think we all accept that. And yet, we're really obsessed with for some very good reasons, with modeling things in systems with more limitations than I think we want to acknowledge. And I think what we really need to do for the benefit of our rare disease patients, as well as the benefit of patients with more complex disorders, we have no idea what starts MS at the outset. And we really don't know what starts even a lesion after somebody has MS. We need to listen to these disorders and try and understand them in ways that maybe don't seem intuitive at the outset. But we are not going to understand these things through mice in many cases. We're going to understand these through human biology. And so, if we then move back to our haves and have-nots, we can look again at some hope here and see that it's possible we can move pieces of these advanced stage boys. We can maybe begin to conceive of some trials and some therapies for these boys. And the same for these boys who were apparently at risk one day. We can imagine a paradigm in the future where, in childhood, if we have a 40% risk of cerebral ALD now, what if we have a low-risk therapy, something like vitamin D, that we're supplementing it? It's not going to cure you, but it may move your risk. It may perhaps it cuts your risk by 20% or 30% or 50% if we dose it right. So so 20% of those, so instead of a 40% childhood risk, you have a 20% childhood risk. These are the incremental gains we can make in this way. And so then to take it back to kind of synthesize it into this is a purely hypothetical model here, but this is where we're kind of like we're looking at here. Basically, if we start with a selective vulnerability in fatty acid metabolism, and in ALD, and we move into this period of childhood that has a very, very high regional tissue demand the brain requires a lot of fuel. And specifically, the areas of the brain, some areas of the brain are not well vascularized, specifically the corpus callosum that is being affected. It's hard to get fuel to that area of the brain. If we think about these as just a supply-demand mismatch, and we understand that the time and the biology are both relevant, so the state of neurodevelopment and the underlying risk factors are both relevant, we can begin to move into this area here where maybe we're just talking about a discrete period of acute metabolic risk. Is there a way we can modulate that period safely? If we don't, then these cells move into localized, perhaps glial activation, necrotic cell death. The cells move in, they scavenge this area. They have to eat the fats in that area to turn them over. And if you're an ALD cell, you can't digest all these fats, and you basically wind up with a set of bones that then crystallize these fatty acids, and then they become a problem for you as a cell. You've got a buildup of these crystals, much like we see in atherosclerosis, but perhaps more severely. And these then perhaps lead to a disruptive cascade that then recruits more immune cells to the brain, increasing metabolic demand, perhaps to the area, and pulling metabolic demand away from the cells that need it, and putting us into this downward spiral of increased demand, supply-demand mismatch, cell scavenging, cell distress, increased signaling, more cells needed, et cetera. So if we've used these other diseases, multiple sclerosis, to identify potentially vitamin D as a target, and perhaps stroke and other disorders to identify cyclodextrin as a therapeutic avenue, and there will be other therapies, I think, like these that we will probably try and test, and maybe we can move towards something at least of a reasonable mouse model for cerebral ALD to get there. But I'll close there. I think we've got time for questions, but I want to thank my lab. We've got a really wonderful team of people in my lab that have done all the computational and bench work. We are running a clinical trial it's a dose titration trial of vitamin D in ALD boys. I didn't show that data here. I want to thank the University of Minnesota collaborators, Paul and Troy in particular, who provided some of the samples we've used in our work. And Christian Doyle is at the University of Arizona. This is the stroke biologist I mentioned, and Patty Mussolini is at Mass General. We've got a number of other collaborators who provided critical samples, and I'll take some questions from here.
Hi, thanks. So I'm curious about the sort of analogy with MS. And when I am a rheumatologist, when I think about MS and other autoimmune diseases, if you look at GWAS studies or Manhattan plots of what are the risk factors, that the thing that jumps out in nearly all of them is HLA associations. So I'm curious, in ALD, does the risk of developing cerebral ALD segregate according to HLA?
Yeah, I think Paul has actually looked at that. Your group, have you not, Paul?
Not extensively, but there isn't anything that's been described thus far that correlates the HLAs with, the onset of disease. So, we may be able to go into it more depth, but at least the initial stuff that was out there suggested that there was no correlation we could find.
You have a small n. I'm also curious, separate question related to the inflammasome activation. Are there thoughts of using biologics that block the downstream effects, IL-1 inhibition or IL-18 inhibition?
Yeah, it's a new idea. In fact, we haven't even published the work on it yet. So, but yeah, I think those will be some of the things we'll be thinking about. We might look at it from even a step further back. So instead of just blocking the immune cascade, I'm thinking about some of the metabolic gears upstream. But clearly both could be reasonable targets for advanced stage disease for boys with no recourse and just a death sentence ahead of them. There are a lot of things on the table. Yeah. I should mention many of the other immunomodulators, cyclophosphamide, other things have not worked, and Troy and Paul and others have tried some of those, and they've been mostly disappointing. Some of the other MS therapies, like interferon beta, which is not much of a therapy, even in MS, had no discernible campath, have not had an effect. But I do think that if we're thinking, at least if we're able to think about specific pathways, we at least increase the potential yield. And the HLA question, I seem to recall Troy having looked at this before, but we do have all the transplant data, and so it is something that is amenable to study. I think Troy said that he hasn't seen any signals, but it's a very good thought. Yeah.
I'm also intrigued by the MS analogy, and it's said that MS is unheard of in Epstein-Barr virus seronegative individuals, and there are also these provocative associations with human herpes virus type 6. I wonder if anyone's looked at the have-a-lots and looked for discordance in herpes virus infection status or not.
Yes, again, I'll point to Troy. Troy is so Troy's looked, I think, at active, I think maybe viral PCRs in the CSF, but serology of viral exposure. So there's another interesting virus, CMV, that actually may have some metabolic effects of its own that actually upregulate very long chain fatty acid production in a way that could actually be meaningful to ALD risk. And so Troy and I were just talking last night also about looking at this on maybe on kind of a broader array of some like a phage display antibody repertoires where we can screen for a wide range of things to at least understand the risk a little more broadly in some of the boys. The hard thing of those is they don't give us an actionable target for things. For the most part, but they at least help us understand some of the otherwise randomness to the process, and that's valuable. Yeah.
So, wanted to follow up on your vitamin D questions related to MS and to ALD. Have the boys, you showed vitamin D levels in kids with CLD. Have they? Is there a pattern in when they were born, as you said before, the latitude, longitude question, yeah, or the vitamin D level or month that their cerebral ALD was identified?
Yeah, the month of birth will be the easiest thing to study, and I've just begun thinking about this exact thing because this is something that will be available in every ALD database. The latitude of birth is a little harder to get at, but we can basically just extrapolate from wherever the person's living now in their first decade of life. The onset is a little trickier because it's such a slow onset, and actually determining exactly when it began may happen over months, and so pinning it down in that fashion is a little trickier. But the other two are tractable, and those are things that we are going to have to look at. It's a fascinating story in MS. It's just some interesting aspects of vitamin D biology that our lab is quite interested in. What happens to you early in life, this is true so much of the time, casts a big shadow over the rest of your life, and vitamin D seems to be one of those substrates that's subject to that. Yeah.
So second question is the analogy to metabolism and the metabolic hurdle. Is there a similar metabolic hurdle in the adrenal gland that we know of, in the adrenal cortex, that is the mechanism by which, since it's a non-inflammatory disease, the mechanism by which this happens to cause adrenal insufficiency?
Yeah, it's a great question. I don't know the answer to it. I wonder if there might be ACTH cortisol natural histories in some way that would maybe shed a little bit of light on that. I don't know. It's a great thought because by age 10, about 50% of boys, it looks very similar to that. So it could be that it's actually kind of mirroring the metabolic demand of the brain somehow. But age 10, 50% of boys are basically adrenally insufficient, and then it tapers off again, much like the cerebral risk does. So there could be something to that.
So there's another question back here, but so for some of our potential collaborators in the room too, I just wanted to point out that, historically we probably see more cerebral ALD than any place in the world, but our experience has primarily been cross-sectional. So the kids get cerebral disease, they come here for an evaluation and transplant or not. We haven't had the opportunity to follow these patients longitudinally to ask these kinds of questions, but now that newborn screening is happening, now there's a cadre of patients that we're going to be seeing and have the possibility to be able to get samples on, for instance. So if people had some good thoughts about what we might want to do with those samples, then let us know, because it's a new age, really, because now we have the ability to follow these kids and potentially pick up things that may be events that are triggers that we couldn't otherwise appreciate. So, sure.
Hey, great talk. I've been always interested in how metabolic environments influence macrophage phenotype, and I'm curious the parallels you're making between like stroke and ALD. In particular, I'm wondering if one, if you think it's more of the local microenvironment in terms of metabolic availability that's influencing macrophage activation, or two, whether it's the macrophage responding directly to neuronal tissue injury. And kind of following that up, I know you showed transcriptional studies already in the context of vitamin D, but have you also looked at seahorse or other things looking at metabolic flux, capacity, et cetera, in terms of what vitamin D can do to macrophages?
Yeah, good questions. We've looked at vitamin D's effects on oxygen consumption and ECAR measures in seahorse, and we're getting different signals based on whether we use the active form, 125 hydroxy or a 25 hydroxy. And I think that is so it's a little hard to know how to tell the story, and it also varies from person to person. We can get consistent results within a person, but it's not always the same from person to person. And so there's a variable in it that we don't fully understand. So we've moved to kind of just in vivo models to try and get at the same questions. And we have a kind of a grandiose theory about vitamin D's function in biology, and that's basically trying to explain its linkage to sunlight as basically providing an environmental cue to the organism about the energy availability of basically carbohydrates derived from UV light and also warmth derived from UV light, such that the organism can synchronize its metabolism according to its environment. And if that is the primary function of vitamin D, it would explain a whole bunch of broader ideas that have been unexplained otherwise in vitamin D biology. But I think the in vivo stuff will I'm hopeful will provide a clear picture. I can see from the literature that the literature suggests that what we're seeing in our transcriptome is more or less reflected in like some in vivo modeling in mice and C. elegans, which is the other model we're looking at. So I'll have more on that later. The in vitro stuff is a little trickier. We can't figure out what the variable is between patients that's producing it, but the general idea is there. Yes. Yeah.
And even just real quick following up on that, I wonder if vitamin D levels in ischemia reperfusion patients, if you see, I don't know if you've seen any clinical correlations with that in terms of long-term brain
This
injury.
is what makes vitamin D kind of a challenging and kind of like that vitamin D insufficiency links to every disease. And so it's almost is a meaningless biomarker at this point because it is. I think there are very few disorders where it hasn't shown us some risk association. So then now really is a matter of finding the mechanisms of why it matters for those diseases and how much of it is epiphenomena of an acute state versus a kind of a preceding factor. Yeah. So we've looked at it mostly from a preceding risk factor to try and mitigate that variable. But in stroke and critical care, every outcome, and I would say pediatric ICU work, there's some neat work coming out. Every variable, bone marrow transplant, I think, vitamin D status is relevant. Now, too much, everything is a poison but the dose. So too much is going to be bad, but there are too little, too much. There is an avenue in between that works. Yeah.
Yes, so a suggestion, has anyone looked at bioavailable vitamin D, not just vitamin D levels? One also has to look at vitamin D binding proteins, and then there's an equation to figure out the level of bioavailable vitamin D. And then my second question is, it seems like there's this bimodal distribution of the onset of cerebral ALD. So what is it about puberty? Because it seemed to be during the pubertal age group that there was a real big decrease in incidence. What is it about puberty?
Yeah, it's a good question. So on the vitamin D availability, this is really the first look, and it's even then a small cohort. So we haven't looked at that yet. We may do that in the future as a next step question. The relation to puberty is, I don't know. In fact, is puberty somehow driving some changes in neurometabolic demand or vice versa? Not clear, but good questions. Yeah. Yeah, okay.
Yeah, so I think we kind of hard stop, so thanks again for your discussion. So, thanks, everyone.
You have been disconnected from the meeting.